Bio. This is Mr. B, and we are continuing on with a new chapter today. So the title of our new chapter is DNA and RNA. So DNA, we have learned about that plenty this year, about how this is responsible for storing all of our genetic information, our chromosomes, our genes, our alleles, all of those things. And RNA might be a new topic for us, so keep that in mind. We're not going to dive into that today, but just know that will come forth in this chapter. So our first section is section 1, and it is titled DNA. So we're going to learn the basics of the DNA, and before we can actually learn about all that, we have to learn about the people who discovered DNA. So there's several experiments that we're going to take a look at that kind of help us understand what DNA is today. So our first experiment was done by a guy named Frederick Griffith, and he discovered a phenomenon known as transformation, which we'll get to in a second. So in 1928, Griffin was trying to learn how certain types of bacteria cause pneumonia. And again, pneumonia is just a disease or an illness that kind of just gives fluid in your lungs, and it's kind of really hard to shake. It's like a really, really bad cold. And he isolated 2 different strains of pneumonia bacteria from mice and grew them in his lab. So he was using mice as basically his test dummies for this experiment in order to see how pneumonia affected them and growing different types of pneumonia. So he made 2 different types of observations. He made the first one being that the disease-causing strain of bacteria grew into smooth colonies on culture plates. So a culture plate is just kind of like a petri dish, if you heard that term before, where you can grow bacteria in order to study them. So the ones that caused disease grew in smooth colonies, and the ones that were harmless grew in colonies with rough edges. So that's a pretty big difference that he noticed right off the get-go. And so after he made these observations, he set up his experiments. So he set up 4 individual experiments. And the first one we're going to look at is obviously experiment 1. So in experiment 1, mice were injected with the disease-causing strain of bacteria. The mice developed pneumonia and died. So it's a pretty simple experiment. You can see over here on the right, disease-causing bacteria being injected into the mouse, and the mouse dies. So that was experiment 1. Experiment 2, mice were injected with harmless strain of bacteria, and these mice did not get sick, and therefore they didn't die. So we can see the harmless bacteria with the rough edges gets injected, and our mouse is still alive. So that was experiment number 2. Both pretty simple, pretty much what we would expect. And so then experiment number 3, this is where it starts to get a little bit interesting. Griffith heated the disease-causing bacteria. So the one that he originally used in experiment 1, he used those colonies of bacteria, and he then injected the heat-killed bacteria into the mice, and the mice survived. So that's a pretty big discovery there, saying that something in that heat did something or another to that bacteria to where it doesn't cause disease anymore. And so the mouse was able to live after being treated with what was originally disease-causing bacteria, but the heat did something. So that was experiment 3. And experiment 4, Griffith mixed his heat-killed disease-causing bacteria with live harmless bacteria and injected the mixture into the mice. So going back, he basically took his bacteria from experiment 3, mixed it with his bacteria from experiment 2, and then injected it into the mouse. However, this time the mice developed pneumonia and died. So that is, something happened here, something different happened that he really didn't expect to see. So we're gonna get to what his conclusion was, which again is the next part after making our experiments. And his conclusion was that the heat-killed bacteria passed their disease-causing ability to the harmless strain. So even though they had been heat-killed, their ability to cause disease was still there in some way, shape, or form, and so they passed it on to our harmless colonies. And this process was called transformation. So that's where this comes in, because one strain of bacteria, the harmless strain, had changed permanently into another, the disease-causing strain. So we know transform, when you transform, is just basically mean changing. So in transformation, it is just the fact that one strain of bacteria is changing into another. And Griffith hypothesized that a factor must contain information that could change harmless bacteria into disease-causing ones. But he didn't know what that factor was. He didn't know what the method for that harmless bacteria becoming the harmful bacteria was. So another scientist came along by the name of Avery, and he repeated Griffith's work to determine which molecule was most important for transformation. And so Avery and his colleagues, they made an extract, which is just kind of like sucking something out and like just a little chunk from the heat-killed bacteria that they treated with enzymes. And enzymes are just materials that kind of break things down. That's what their job is to break things down. So these enzymes destroyed proteins, lipids, carbohydrates, and other molecules, including the nucleic acid RNA, which we haven't got to yet, but we will, and transformations still occurred. So all of these proteins, all these lipids, all these carbohydrates, and other molecules were destroyed, and transformation still occurred, so we knew that none of those were responsible for transformation. So Avery and other scientists, they repeated the experiment using enzymes that would break down DNA. And when the DNA was destroyed, transformation did not occur. Therefore, they concluded that DNA was the transforming factor. So when they were able to destroy DNA, they were able to keep transformation from occurring. And therefore, we knew DNA was responsible for transformation. So what is the relationship between genes and DNA that came from all this? So Avery and other scientists discovered that nucleic acid, DNA, stores and transmits the genetic information from one generation of an organism to the next. So the combined work of Avery and Griffith is kind of how we come to know what we do about DNA today. So that is it for this section of section 1. Let me know if you have any questions.